So I should say at the outset that I'm really not an expert in bacterial cytokinesis, and whatever little I learned, I did over the last few days with Srinivasan. So, so there has been a few questions from the audience about, there are a few questions that I think maybe I can try answering. So I'll basically try to follow up the some important modeling papers in this area in a more or less chronological order. Okay, and so this is the first paper which I thought was reasonably important. Sorry, not this one. Took the wrong one. Another PNS. Yeah. Yet. So here, I mean, so what they basically do is to look at the, basically model the cell wall as an elastic sheet which can be deformed elastically, and then imagine an Z ring on top or under it, which is trying to constrict it. So, and their idea is that basically by using information about the elastic properties of the cell wall, which they have gathered from earlier literature, from the Young's modulus and so on, they'll try to estimate the kind of force that is required to deform it to a shape that it resembles before cytokinesis starts. So that is basically the setting. And it's essentially a theoretical paper. And as I said, the output of the paper is they arrive at an, so I think I can also write down like what are the essential parts in the energy that they are writing down. So they basically have this cell that is about to deform, and then you have the Z ring here, which they don't really model in detail, but only as an object that is generating a radial contractile force. So the force itself is not modeled in detail, and they don't talk anything about the mechanisms that go into generating this force. But you just start with the total energy of the system, that is the, I believe it's the bending energy and then the stretching energy, all for the cell wall itself, and then the energy, the contribution to energy from the turgor pressure inside the bacterium, and finally the E ring, which is just modeled as minus F times R or something similar to that. So F itself, F is what they're trying to compute by finding the minimum of this energy, and then basically trying to figure out what F should be so that the kind of deformations that has been observed in experiments can be reproduced. So that is the idea. And the sort of bare estimate that they arrive at is actually quite high, and they first argue that the kind of deformations to be produced, F has to be of the order of 400 piconewton, which is certainly quite large for any sort of reasonable cellular machinery to generate. And then they bring in the what has already been discussed by Srinivasan, that the peptidoglycan by the layer actually forms and reforms over and over again. So then the argument is that the Z ring need not generate so much of force, but rather once it generates a small force and creates a furrow, maybe the layer will go away, new layer will form on top of it, and therefore there is a dynamic sort of process that is going on, which will allow the Z ring to sort of cleave the cell with less than 1/10 of this force. So the sort of absolute minimum that they claim that is required is of the order of, let's say, 8 to 10 piconewton. Yes, Srinivasan, is it a reasonable estimate? Because I don't have the actual number in my head. Yeah, it is there, right? Right. Okay, fine. So that is basically what is summarized in this paper. I'll just see if there is anything else that we need to go through. Yeah.
There'll be a lot of other parameters in the bending, stretching, and all.
So they have basically got estimates of most of this from the existing literature, it seems. That's what I figured out. Of course, as I said, I'm not really an expert in this area, but in fact, I have given a list of these parameters in a table, and that's a good question. No. I'll just see. I think it's an energy minimizing problem. I don't remember they do also have dynamics. In the appendix, they do also discuss dynamics. So I think it's a dynamics problem. And for the dynamics, they use this energy. So it's an overdamped Langevin dynamics that they use, correct? Right. Yeah, so this is basically the primary conclusion. The role for the Z ring, as they postulate, is to generate a small force to establish the direction of the cell wall growth, and it's to concentrate the growth. I think both these aspects Srinivasan has already discussed in his talk.
So if you're doing dynamics, then of course there's a fluid inside, right? Inside that bacteria, but the fluid is incompressible. So somehow you have to expand it in the lateral direction or something like that you have to do, no? Yeah.
I believe it is done. I believe they have dynamics along the Z direction as well as the radial.
No, but do they put the incompressibility of the fluid? Volume constraint?
That's too detailed a question for me.
Okay.
I'll have to look at that paper. Okay. Yeah, I understand.
Just from that figure, it looks like it's been done.
Yeah. Right. Right. So if you want a list of the parameters that they have used, I think they have a table somewhere around. I can just sort of quickly. I think this is list basically all the parameters that they have used. The references I'm not seeing here, but I think they are scattered all throughout the text.
What happens in the bacteria itself, in the experimental situation? Does it grow in the lateral direction while the?
Yes.
It does. Okay.
Yeah, because it's rapidly growing, at least in the rapid. So when bacteria are growing so rapidly that the second division site is already established even before the first constriction is completed, and the bacteria grows, right? That is what, not bacteria.
To conserve volume.
Is it conserving volume? Yeah, still the ring volume, the volume of the bacteria. So I'm not getting.
Okay, so any further questions on this that I can answer?
So the ring position, that's put in by hand, or that's something?
Here it's put in by hand, yes.
Nothing spun.
They haven't done anything.
But since they have dynamics, probably they could try to put the ring away from the
The ring itself is not modeled in detail, as I was saying.
I see. It's just a force that is there.
So.
From the images,
Yeah.
it looks like they do put it
I
offset.
think they may have, but still static. It's not really moving in any way. So they may have tried it to the other positions and just tried to see how their conclusions change. So, okay. All right. So, so the next two sort of more. So the next two papers essentially deal with mechanisms for generation. Yeah. Please.
So it's nice that they get an estimate. The estimate of force is a reasonable estimate,
Right.
the 8 piconewtons. But for that, they have to have a rate of chemical synthesis of the cell wall.
I think they have used it.
They've used it, but
They have used
and that is also reasonable. They can compare that. Huh?
There is a table.
So there's a table. They must have compared that to
I do
experiment.
not know the source of
Yeah.
it and follow it up.
Yeah,
But yeah, they have used a number. All right, so this is a, sorry, not this one again. I am always clicking the wrong files. Yeah. Sure. So the next two papers I'm going to discuss actually are like models for how a Z-ring, which has whose dynamics we know a little from experiments by now, generates a contractile force. Okay, so I'll basically start with trying to write down the essential physics of it, of which, of course, more meat can be added later. That's what these people have also done. So the essential physics is essentially just bending energy of you. So as Srinivasan was saying, after hydrolysis, these filaments acquire a spontaneous curvature. So let us say you have a GTP filament, let us say, prefers to be more or less straight, whereas one which is hydrolyzed prefers a bend conformation with a rate. Is it too small? Is it okay? Yeah. Right. So this is my filament, which is almost all GTP bound, and then after hydrolysis, it becomes more GDP, and then it prefers a bend conformation with a radius of curvature, say put not approx 25 nanometer, right? Around 25 nanometers. Right, and so let us say we have some sort of a ring-like structure. Let us not worry too much about how this was assembled in the first place. So this need not be a continuous ring, but for the time being, let us imagine that it is something like that, that has kind of formed at the junction of the two future daughter cells. So I think I've sort of, maybe the geometry doesn't look right. Yeah, so this is the ring. And let us say at a certain point, it has a radius of r. Of course, it is being continuously disassembled and made again, and there are like layers and all that, but let us think of it as a semi-flexible polymer whose bending energy can be written in a way similar to what Madan had written down for membranes. So I'll write a bending modulus, and then there is an integral over the entire perimeter of the ring, which I'll write as ds as being some sort of arc length, which I measure from a reference point. And then you have the difference of curvature. So this is the original, this is the local curvature, which we take to be the same everywhere. And then, of course, now we have two sets of filaments, but let us imagine for simplicity that we have only a single filament that has, say, a spontaneous radius of curvature everywhere. So that is something like a mean field assumption. And then you can, this whole thing is constant, can be taken out of the integral, and for the energy, then you get something like 2 pi r into 1 over r minus 1 over r naught squared. You can just expand it. You can forget all the constants. So this is proportional to r times 1 over r squared plus 1 over r naught squared minus 2 r naught, of which the only r dependent parts will be, let's say, 1 over r, and then there is an r over r naught squared. The last term becomes a constant. So let us sort of take it from there. And if you now plot that energy as a function of the radius, you see that for small r, the first term is going to dominate, and for large r, the second term is going to dominate, and it actually predicts a minimum. So, and that minimum, of course, has to occur at the spontaneous curvature itself. So at least now see that from the energy minimization point of view, there is sufficient motivation for such a ring to shrink, even if there are no other details put into the problem. So starting from a given radius, and now the ring will, at least from this argument, it is going to try to shrink spontaneously until it reaches a certain radius that corresponds to its spontaneous curvature. But of course, this is not an intact ring. It is actually made up of many small filaments which are being continuously remodeled again and again because of treadmilling and all that. But at least we can get an estimate of the force from here by taking the derivative, and the force will be largest when the radius is the largest, and the starting point it's like 1 micron. And I don't have the numbers here, but one can actually take the derivative here and find the radial force minus dE by dr, and then that can be easily calculated, and I think it turns out to be of the order of 10 piconewton. So therefore, certainly in modeling a problem like this, so one of the sort of questions that we would start with is the magnitude of the contractile force. The second would be, let's say, the time scale of constriction. And what else could be there? Maybe the, the structure or organization of the ring. Etc. etc. So the force actually gives, I mean, that's like one of the first questions that a modeler would try to answer. Okay, and so this is a very sort of very basic zeroth order kind of estimate that we have done here. And so what Anirban and his student did in 2008 was to actually simulate a computational model, of which I'll go through some of the details here, because I believe, and people can correct me if I'm wrong, that this is, I think it's the first paper that tried to model the constrictive force generation by the Z ring, right? Am I right, Srinivasan? Yeah, right. So essentially the ingredients of the model, as you can see here, as you know, there is the curvature energy similar to what I've written, but you know, this is more detailed because now our polymers have both GTP as well as GDP, and therefore the spontaneous curvature is not a constant across the filament, right? So either one option is to use a mean field approximation, the other option is to do it by computation, and these people use the second approach. And now there are, of course, that's not all. Now, he also puts in, they also put in like other energy scales in the problem, like the polymerization energy. So Anirban's rings are actually, they're actually planar rings, so there is just sort of one layer here, so it's not really a barrel. So they used a simplifying assumption for that. So he basically has a set of rings, concentric, which is being fueled by GTP from this side. And there will be more GDP towards the end, and GDP polymers, GDP monomers are unstable and they would tend to disassemble faster. So that is the idea, and that is how treadmilling is generated. So if you sort of look at a cross section of this, then let's say I put a filled circle as GDP, open circle as GTP, and then I have, strictly speaking, it's curved. So GDP is being, sorry, GTP bound monomers are being put in from one end, GDP dissociates from the other end, which produces treadmilling. And then so there is an energy for polymerization. And what is coming inside are actually polymers of all sizes. So there are monomers as well as dimers, oligomers, everything is going in. And I think so the two additional elements of energetics that he has included is, one is the energetics of polymerization, and then there is also lateral contact between two adjacent rings, which these others sort of implement only between two GTP polymers. And therefore, wherever you have more GTP, the rings will be more stable. And so towards the outer, where there is less of GDP, because they don't have like GTP coming in there, the rings will be unstable. And therefore, as a result, the ring will continuously contract. Also because of the energetics. So it's a combination of all these things. So, and what they did, so the kinetics, of course, is completely governed by the turnover time of FTSZ in this model. Okay, and I think so then the simulations, they essentially predicted the time scale of shrinking of the ring, for which given a figure here, which is roughly 10 minutes, which matches with the experiments more or less. There is no direct estimate of the force other than the kind of simple analysis that I have given here. In fact, they predict a rather wide range for the force, ranging all the way from 3 to 300 piconewton or something. How do they?
How do they contend with the fact that the volume, that the fluid inside is incompressible?
They don't model the cell in any detail. The model is only on the ring. So this is a contractile ring.
But it's squeezing.
It is squeezing in, but the fluid is going away, but the other part of the membranes are not being modeled at all.
No, it doesn't need to be. But the point is that it's a confined medium and it's an incompressible fluid.
Absolutely.
So whatever force you apply, you won't be able to squeeze it unless you grow in the... So they just assume that it grows in the...
Nothing prevents their cell from growing in the other direction because that cell is not there anyway. They don't have it in the model. So
you're saying that they don't...
is only on the ring.
So they don't bother about that. Yeah.
So the elasticity of the membrane of the cell wall doesn't enter the picture at all.
Elasticity is just that. So I guess you're saying that the confinement.
Yeah, but the size itself doesn't enter the formalism.
No, but the balloon is.
Etc. What is being produced inside the synthesis which is going on. So the net surface area is also expanded.
So that's the rate. Thanks to maybe set by how fast you had. It's an important problem. A lot of people are aware.
But in this case, there is really no change in surface area because they only have a ring which they are shrinking. Right? Okay, so I'll try to quickly go through the other two papers. Let me see if there are any other important points to note here. Yeah. It's a good point to stop. Come back and absolutely. Your take. Yeah. So we'll shall you announce or you will announce? Okay. So we'll stop for coffee break here, and we come back after 15 minutes. Okay. 3:30. Okay, welcome back. So we'll continue the commentary come discussion. So the third paper that I wish to mention is this paper from Eric Sittenbaum, this collaborator. So mostly the setting is similar to what was discussed in Anirban Sain's paper, except that they have added some more details and made the model a little more elegant, so to say. And so it is primarily a mathematical study rather than computational. And they've got they get explicit expressions for the force and so on, but more or less the terms in the energy, etc., are very similar to what I had written down earlier. So there is the bending energy of the filament and the GTP hydrolysis of the filament is included in a mean field manner by assuming a stationary state situation and demanding that a certain fraction of the monomers has to be in the GDP state, certain fraction has to be in the GTP state. If it is in a GTP state, then this is the contribution to the spontaneous curvature. If it is GTP, there is another contribution. And I think some energy for lateral contact has again been included to stabilize the ring. So that is basically the idea. And so after that, essentially, it's a minimization program that they're doing. So this cartoon sort of summarizes the essentials of it. And basically, the only additional feature is that instead of Anirban's planar annular ring, you really have a barrel-like structure in this model. But I don't think that actually changes the physics all that much, except that the direction in which the GTP is entering the system is now lateral rather than from radial as what was there in Anirban's model, or mostly. So if you wish, I can just go through a little bit on the detail, but I don't think there is actually much new physics that is being added here. So there's a rate equation for the probability distribution of the length of the filaments in the ring, and then you have a supply of filaments from the cytoplasm, which follows an exponential distribution. And then there is a second ordinary differential equation that has that basically describes the kinetics of the fraction of monomers in the GTP or GDP state, as the case may be. Okay. Right, and then the stationary distribution of the filaments, the length distribution of the filaments in the ring is also found to be exponential with the same length scale as originally included. And then they get an expression for the force, just by differentiating. But something I found a bit surprising was that the force actually has a different behavior compared to what I had drawn there from the really elementary analysis. So instead of, oh no, this is actually the force. That is the energy. I'm so sorry. Fine. Never mind. Never mind. So this is, no, actually I think in the model that I have written, the force, I had to calculate the slope. So sorry again. I didn't calculate the force yet. So anyway, so this is their expression for the force as a function of the ring. And as far as these authors are concerned, basically the only point that you're interested in is whether in what regime of the ring radius they can produce this minimum 8 or 10 piconewton kind of force, and they found that it is appreciable. So starting all the way from the cell radius up to almost, I believe, 10 or 20 nanometers, they get a force that is above the requisite minimal force required to constrict the cell wall, right? So that is basically the conclusion of the study. Are there any more questions? Yeah.
So what is the point of this? I mean, now they have some dynamical equation. Can they get some dynamical character? I mean, otherwise it's just energy minimization.
It's essentially only an energy minimization. So that's what I said in the beginning. There isn't any essential new physics being added here compared to the previous model. Nothing that I could find anyway. There is just a little more detailed treatment of the length distribution of the filaments in the ring and all that. And the turnover rates have been put in explicitly and all that. But that might be there in the previous problem also. I'm not immediately able to remember.
No fluid, no incompressible, nothing.
Nothing.
Manoj, I have a question not directly related to this model, because these filaments undergo treadmilling, so they're polymerizing and depolymerizing. So there is a K on and K off.
There's a K on and K. In fact, it's a K on and K off that determine the kinetics of the ring constriction. The energy has nothing to say about that.
But my question is slightly unrelated to this. So what prevents the filament from breaking anywhere in the middle? Why is the polymerization, depolymerization only at the ends?
Why can't the GDP monomers, for example, just drop off from the middle?
Yeah. In the case of actin, the argument given is there are two filaments.
That's right. Yeah. I understand. I understand the question. But that seems to be a very broad question, not restricted to the model itself, right? I mean, Srinivasan, do you have an answer to that?
It might be just possible that the longitudinal contacts between these filaments are still strong enough to hold it, even with the GDP. Even though it's a constrained bend filament, the longitudinal contacts may be strong enough to hold with the GDP. If that is there, I don't know exactly if it is really strong enough to hold.
One at the end, the energy is halved. That's the idea, right? The one in the middle has two neighbors to bind to. The one
so
there is only one. So maybe I don't know.
So at least the top and bottom subunits are there. They can hold the filament. This subunit which is GDP hydrolyzed still in place.
Or unless when you have two neighbors, then the conformation is different, and then the sort of K on K off is not the same. Is it possible?
Anyway, K on operates only at one tip. K off operates only at the other tip. So and there is, I believe in none of the models that I have seen, there is a K off from the middle. There can't be a K on there anyway. So I do not know if that's a point you are trying to make.
No, my point is that the probability of a dissociation happening in the middle should be same as the probability of a dissociation happening at the end.
Now, what Srinivasan
when you
was
have
saying,
only a one linear filament.
Yeah, but a monomer in the middle has two neighbors to form bonds with, whereas the one at the end has only one, right? So that actually reduces the energy of bonding by half. So maybe there is a higher probability of rate of dissociation from the.
You can think of the same thing for microtubules or actin, for example.
But actin, there are double filaments, and this is the reason why it's given that only at the ends, because at the ends the contacts are less for the monomer.
No, but the spontaneous actin filament breaking in the middle is also highly unlikely.
It's highly unlikely because of the reason that there are two filaments going around each other.
I guess that's the same thing that is.
Yeah, but there are longitudinal bonds, right, between the monomers in the filament itself.
If you have multiple filaments side by side.
No, within a single filament, you have a monomer here, monomer on top, monomer at the bottom. For the filament at the end, the monomer at the end, there is only the monomer at the top. So it has only one bond. I don't know, it's only a qualitative argument, but I'm just guessing that that might be a reason. I can't think of anything better. Right, so any other questions from these models? Because I'm going to switch to a somewhat different paper. That is the last paper that I'm going to review. Okay. So the last paper is somewhat more recent, 2016, from Erwin Frey's group in Munich, and this is more a sort of active matter philosophy motivated paper. So, and the focus is not so much on the constriction force, but rather on the likelihood of ring formation when you have a set of FTSZ monomers which can hydrolyze, etc., and which are now treated as active particles because each filament also has a treadmilling property. So the idea is that you have a collection of such sort of small or micro, I don't know if micro is the right word, filaments in the system. And basically the model is that you model, so when you have a filament like this with GDP at one end, GTP at the other end, we know that GTP is being added from here, GDP is released from here, and there is treadmilling happening, and the filament, if you look at it from outside, gives the impression of moving forward on the whole, right? So then what these authors did was to approximate this as an active particle, which is now a point particle. So it has a sort of self-propulsion velocity, which essentially matches with the observed velocity of treadmilling of the actin filaments. But the one modification that they made is that while, so what they proposed is that instead of sort of moving in a straight line, even with a single monomer, it actually follows a curved path. And the radius of curvature of this path is essentially the radius of curvature of the GDP form of the monomer, of the filament rather, right? So that is the model. And then what they did was to have a collection of such particles of variable densities in a two-dimensional plane, and just let them move around each other, collide with each other, and the only interaction that was assumed was hardcore repulsion, nothing else. And then like what sort of patterns, spatial patterns are generated. So the focus is more on whether like the vortices, the swirling vortices that were observed in these experiments that, yeah, Vaibhav? Yeah. So it's not coming in explicitly. So all that they say is that when a polymer when a point particle moves forward, its natural path is going to be a curved path, not a straight path. Right. So if you wish to see the details, I think it's somewhere here. So this is what it is going to do. On the left hand side, you have sort of a cartoon of real filaments, polymers, and the right hand side, the effective active particles. But the important point is the only interaction is hardcore repulsion, nothing else.
So when they go around in circles, they always go the same way? They don't go the...
I think the chirality is built into the particle. The particles are asymmetric. They look like Janus particles to me.
No, it's not a chiral question. I'm just asking if you look at all the arrows, they're going all, whatever, clockwise.
Yeah.
Right? So there's no, all the particles go the same, okay.
Yes.
If you really had a mixture, probably, I don't know if you could stabilize such things.
I don't know what mixture of what?
Mixture of
Chiral, I mean, okay, clockwise.
Clockwise and anti-clockwise going particles.
Yeah, that's an interesting question because I think there are mutants which actually follows swirl the other way, right? I do remember seeing a paper.
No, that is only because of the membrane binding.
Oh, the membrane binding, and there is no membrane here anyway. Yeah. Okay, it is not a mutant. All right, so I'll just go to the results because, so there are actually two parts of the paper, which is a very nice paper. So there is a numerical part and there is a nice theoretical part. Theoretical part I didn't go through in detail, but I'll just show you the results of the numerical simulations that they have. So essentially the objective was to see if they could reproduce the results of one of these experiments that Srinivasan had mentioned. I think it was not the loose Mitchison one, but maybe the one after that, I forget which. But anyway, so the idea was that it's purely with very simple interaction with no sort of more details of biology coming into the picture, can you still generate patterns similar to what were observed in experiments with the FTSZ? And so this is the result. So, essentially the qualitative part of it matches with what were observed in those experiments. So as you increase the density, the average density of monomers on the surface, you see the system going from a sort of disordered liquid-like phase to an ordered phase where you have a lot of vortices forming spontaneously. And these vortices, by the way, they don't have similar radii. I mean, I think there is a distribution of radii, but I didn't really see any plot here that actually gives the distribution. In fact, not even a mean of the radius is given. Maybe, I'm guessing, because they couldn't really characterize the vortices very precisely. So they had to resort to some sort of a correlation function to actually sort of deduce the existence of these vortices and structure formation. And the interpretation was that if the minimum matches the spontaneous curvature, then it's a disordered phase, and if it's sort of slightly or more than that, then you get to the vortex phase, and then much beyond that you get to a bundle phase. So broadly speaking, these observations match what were observed in the experiments, and that was really their goal. Okay. And there is a sort of very nice Landau-Ginzburg kind of theory that follows that, but which I will not go into detail. And so that is where I don't think I've actually highlighted anything else after that. So that is where I'll stop essentially, and I am happy to take any more questions that anybody may have. This. So if not, I'll hand over to Srinivasan to continue his talk. Okay.
So this is the second part of the talk, cytokinesis, the DNA segregation. Again, here DNA segregation, I mean the simple circular plasmids. Chromosomes are circular, but they are huge. Plasmids are the smaller molecules for which DNA segregation is more understood in bacteria. So most well understood is the system called as RMRC system, where if you go back to the first talk that was there, Kautsev's talk, that there is a kinetochore, there is a DNA element, a centromere, then the kinetochore and a polymerizing filament that just segregates the plasmid. So you had like hundreds of proteins there and assembly of kinetochore, centromere, histones and everything. So here it comes down to just 3 elements. One is the DNA sequence itself, which is a repeat of 10 to 15 base per unit, and you can have n number of repeats. It varies in large number of bacteria. And a protein that binds to that particular itron or the repeat in the DNA, and another protein that forms this polymer. And on the DNA sequence, they occur in these forms that these are the DNA elements to which this protein marked in green binds. And this is the filament forming protein that binds to this element and then pushes the plasmid away. So this video from Dyke Mullen's lab actually explains it very well. So there is a plasmid, this is the DNA element, this part to which this green element is brown, this orange one. And then the polymerizing protein polymerizes from these. And as long as there is no other plasmid, there is dynamic instability, like in case of microtubules, the ATP cap is lost. But as soon as there is another copy of the plasmid, they are stabilized at both the ends, and insertional polymerization, like actin, mediated by the formins, trigger polymerization, and they are just pushed towards the cell ends. And the cylindrical cell will divide in the middle, and it ensures that the plasmids are inherited by both the daughter cells. So it just keeps pushing the plasmids away from the middle, nothing else. It's not cell cycle regulated, nothing to do with bacterial cell cycle. You come to the middle, as soon as it comes, you just start pushing and always keep it at the ends, and cell will divide in the middle, and I end up with 2, each copy on either side. And these kind of segregation systems are found in plasmids which have a single copy number. These are not found in multi-copy number. So if you have a large number of plasmids like mitochondrial division that Vaishnavi was talking about, you have large numbers, you just segregate randomly, so I get plasmids enough in both the bacterial cells. So this is one kind of molecule that is very well understood on plasmid segregation, how it polymerizes and what are the structures. And the other class is the Walker A type of ATPase, as I told you, a separate class of cytoskeletal proteins that are there in bacteria. So one of the earliest hypothesis of DNA segregation was again given by Jacob and Monod, that the DNA somehow binds to the membrane, and the cell wall elongation and the membrane synthesis is just growing as the cylinder is growing. The bacteria is the DNA is attached to this membrane, and that also segregates along with this. So the growth of the bacterium leads to the chromosome segregation. But later there are several models like the spar A, which is an ATPase or ATP binding protein, forms a gradient in the cell. And the molecule that is to be segregated, here the origin of the replication, follows this gradient slowly, and this triggers ATP hydrolysis and kind of pushes the gradient away from this part and eventually gets to the other end. Other models have been like filament model, I'll come to this slowly. Filament model or a Brownian movement which just captures these chromosomes on the ATP and they kind of move in one direction. And there has been more recently a DNA relay model where Christian Jacobs Wagner's group has proposed that the elasticity of the DNA itself is important for this process, and that plays a role in directional movement of these origins. But this particular case is for the chromosomal DNA segregation itself in bacteria, and in this particular bacterium, Caulobacter crescentus. The other cases are more general for plasmids. And what are these proteins? These belong to the same set of proteins that position the ring in the middle. I talked about the FTSE ring. So how is FTSE ring positioned in the middle? Through these three component systems. Again, membrane binding protein, which is marked in green, which binds ATP. It is the same family of these proteins that are also involved in this plasmid segregation. So here this protein kind of binds membrane, ATP binding, and it stimulates this. And there's another protein that stimulates the ATP hydrolysis, falls off from the membrane, and this protein keeps oscillating. And in vitro, you can recapitulate just these two proteins, the min D and the min E, like wave fronts, and how wave fronts move, and the other movie is just, they form this vortices kind of thing on the planar lipid surfaces. So this protein is a membrane binding protein, and this kind of stimulates the ATPase activity of this, and with these two proteins, you can get these kind of patterns in vitro. So the plasmid segregating protein also belongs to the same family of proteins. And I really love this video because it is just completely synthetic. These are not bacterial cells. These are just micro patterned surfaces on which lipids have been coated. And this min D and min E have been just filled into this one on these lipid cavities. And you have all kind of scales of lengths and diameter and everything here. And you see the same kind of oscillations that you will see in a bacterial cell without anything. So coming back to the DNA segregation, this protein that belongs to the same family of this initial models were that they form an oscillating foci, just like this min D protein, and that kind of oscillating foci segregates the plasmids. But later models came that there are filament-like structures visualized in the cell, and there is a filament on the nucleoid that kind of takes the plasmid and involved in plasmid segregation. Then there are gradient model came up that there is a gradient that can be visualized of this protein itself inside the cells by super resolution microscopy. That gradient drives the polymerization. And this is the DNA relay model that I talked about. And to explain this, then there was a diffusion ratchet model or a Brownian ratchet model where this plasmids are moving on these chromosomal particles, where the protein is bound on the chromosome of the DNA, chromosome of the bacteria itself, and the green particle represents the plasmid. So binding to these chromosomes is just moving in a diffusion ratchet model. And more recently, super resolution microscopy and using 3D visualization. And one of the questions that was there is what is the limitation? So what they utilized is super resolution combined with something called as multifocal plane microscopy, where at the same time you can image all the foci. So that kind of, you don't need to go through the C-section and image one by one. You can just image 25 sections at the same time. So they used this kind of microscopy to say that the entire partitioning complex is, I just read up yesterday, and no, not spin light also. It's like they say that they have multiple detectors, and as there is something called as multifocal microscopy. So you get the detector light, and then you have built different tube length of different things with different detectors. So essentially you will, if you want 25 frames, I think you need 25 detectors. And all the focal planes are now imaged. So only yesterday I read about this multifocal planes also. In this they had used such a microscope. Sorry, again, I had individual slides for this, so I had the references. I completely forgot to put the references, the original references in this. I had it in my mind, I completely forgot. I must apologize for that, so I'll put up these references. So the most famous among these has been this diffusion ratchet model and a lot of modeling done. And one of the idea was that if you have, so the red is the plasmid bound to this protein, and this is the ATPase molecule that is bound on the chromosomal DNA. So the idea is that they bind there, stimulate the ATP hydrolysis, release these, and then given a confinement, they would eventually move in a directional motion. And to recapitulate such a system, what they had done is they used a magnetic bead here, held it close to the surface on this glass cover slip with the DNA molecules loaded here. With the magnet, they can always hold the bead very close to the surface to artificially create that confinement instead of putting a membrane there. So that's what they did there, and they could find directional motion. However, none of these could recapitulate the complete 1 micron directional motion that is seen in vivo. So
what breaks the symmetry of the...
When two are close together, there is a depletion zone. And then these have more affinity to this side, but why they are not coming back after they start taking off? So
some gradient of...
that is the gradient of ATP model, but it then goes back to the gradient. So what is this Brownian ratchet model? In the next model, they have this, what they have done is they have done a 2D modeling of this. There's a completely modeling paper. This is the planar chromosome that they have taken, binding sites, and the green is a disk representing the plasmids, and they have marked some kind of interactions here and the K on and on off and binding to ATP. They have an affinity for the ATP bound molecule. So this is, all of this is the nucleoide bound molecule. These have affinities for these. And as the ATP is hydrolyzed, still how is this directional thing coming up as something which I have to understand, even I have not understood this completely. Why is this directional motion coming from? But again, it's not very, it doesn't go as far as the 1 micron or 0.5 micron seen in the bacterial cell, not more than a micron. So this being the question, this is the current scenario that these are either on the surface. All these models were based on the diffusion ratchet model that the plasmid is segregating in between the plasma membrane and the nucleoid surface. It's all happening on the surface of the nucleoid. But the super resolution microscopy completely took it to a different level, that everything is happening embedded into the nucleoid of the chromosome, and there are high density regions inside the chromosome, and all these plasmid dynamics are happening within those high density regions. So still they could reconcile with the diffusion ratchet model that these high density regions are the regions where the ATP bound molecule is present and it is able to diffuse through that. Coupled to the chromosome segregation, it is also hitchhiking on the chromosome. So that is the chromosome hitchhike model that they have. So but what are these models, which of them are true, or probably aspect of all of these are happening in this plasmid segregation is one side. But another thing that I was struck with, that earlier reports on this plasmid segregation, when they first started studying, all of them indicated that F plasmid, whatever I'm talking about is this plasmids are F, and this P1 plasmids use this par A kind of molecules. They partition into anucleate cells as well. So if they were so stably associated with the nucleoid, how do these plasmids get into the anucleate cells? How are they stable? And the copy number is also maintained in this MUGB mutants where you can generate anucleate cells. So the MUGB mutants would look something like this. So you have a cell, both the nucleoids will be in one cell, and you will form a septum here and you will get anucleate cells. So if the plasmids are so tightly coupled with these nucleoids, how do they get there? And copy number should eventually increase in these cells. That didn't happen. They were still maintaining a single copy number. So, and the second thing was that biochemically it was shown using spheroplast and other spheroplast from bacteria and extracts that SopA is, a small fraction of SopA is membrane associated. So this kind of led us to think that maybe there is something more to this thing. And the reason being that whatever be these models, all these models, what they invoke is that there is a time delay between this ATP binding or this release from binding back to the chromosome. So that is where the asymmetry probably comes from, or the symmetry breaking comes from. So as soon as they're released from the nucleoid, they cannot rebind the nucleoid immediately. There should be some kind of a time delay between that step. That is an absolute essentiality. And one of the ideas that the time delay is coming from is that RA can bind ATP, but to bind to the nucleoid, they need some kind of conformational change that is needed, and that is what delays that immediate binding back to the nucleoid. So that's the idea that is currently existent. What we wondered is that if SopA is actually membrane bound, can that also act as a time delay? But is SopA really membrane bound? So that was the question. So what we, at least from this program that amphipathic, which predicts amphipathic helices, you can see in this SopA, this is the F prime SopA, where you can have an amphipathic helix at the C-terminal tail. And these are the other, just to, even as, although it is predictable program, I just use other molecules where this is a closely related SopA which has an amphipathic helix. This is the FTS-A which I talked about, which has a membrane binding target. So that is also an amphipathic helix. So all of these can take the protein potentially to the membrane. And there's a distant par A which cannot bind to the membrane. It doesn't predict any amphipathic helix. And you can see a nice hydrophobic surface on one surface of this helix in this SopA protein. No, septin is more, it is more thought like FTSZ, like GTPase itself, and this has not been thought about. Yeah, so yes, so that, although not related to septin, MIN-D apparently has a membrane curvature sensing motif in itself. It's a membrane binding protein and it can sense the curvature. This one I'm not sure. So what we did is we took this amphipathic helix and started mutating these residues to see what happens to the protein. Normally you would see this protein bound on the nucleoid as that is the major binding thing. But when we mutate these hydrophobic residues, they forming these cytoplasmic rods. So that, and these images are strikingly similar to what would happen in FTS-A if you had to take off this hydrophobic phase. It would not anchor to the membrane and they form these polymers in the cytoplasm. So that again strengthens our view that maybe these proteins are membrane bound. But when we took only this piece of the C-terminus and anchored to a GFP, we didn't see any membrane binding, and this is the positive control where the MIN-D membrane binding portion, when we fuse it to GFP, you can clearly see a membrane binding. So that was kind of disappointing to us. And even with FTS-A forms these nice rings and cells become filamentous only when it binds to membrane. When you replace the FTS-A C-terminus, the membrane bound portion, with this SopA, the par A C-terminal portion, it doesn't form filamentous cells. Of course, it goes to rings, but even without the C-terminus, it has some affinity to go to the FTSE rings. So that was kind of disappointing, but when we check the membrane fraction biochemically, we can see that this has some equal proportions in soluble fraction and as well as in the membrane pellet fraction. And this is of course GFP, nothing there in the pellet, and this is the MIN-D, a majority is there in the pellet fraction. So probably in these images they are also membrane bound, but since they are also cytoplasmic, I simply cannot distinguish by imaging if they are in the membrane or not. Sufficient amount is not there in the membrane. So there is some proportion that is always there in the membrane. So we can at least say that the C-terminus does have a potential to bind to the membrane. And this is work done by Sakshi in collaboration with Anand at IISc. And any questions on this slide? I don't know if she's here. She was in the meeting, but now she is not here, I think. So she took this C-terminal peptide and she kind of MD simulation with the membrane. How long does it take, and does this membrane does this peptide have a potential to bind to the membrane? So she could show that within 50 nanoseconds or something, you could get this peptide embedded into the membrane. It definitely has some membrane binding potential, and these are the same residues that are close to the membrane. So these indicate that this alanine tryptophan, these are close to the membrane, and this is the tryptophan that we mutate and it comes off the membrane. So what we can also show that these mutants do not bind to the chromosomal DNA. They lose their ability to bind non-specifically to the DNA. There is a simple EMSA by just agarose, it doesn't require any extra. You just take any plasmid, wild type will bind to the plasmid and it will show a shift. But these mutants which are in the hydrophobic residues do not show any shift. They have lost their ability to bind to the non-chromosomal, the chromosomal DNA as well. And they also do not maintain the plasmid. None of these mutants maintain the plasmid. This is the ATP binding mutant, ATP hydrolysis mutant, and these are this hydrophobic residue mutants. So definitely they play a role, these mutants do play a role. And what we are currently thinking is this is a schematic. In a biology talk, I would have tended to call this as a model or a working model, but it's not really a model at all, it's just a schematic. So in the case of wild type, the C-terminal tail has the ability to bind to membrane. It binds to membrane. Upon ATP binding, it dimerizes. And upon dimerization, there is some kind of conformational change that allows it to bind to the non-specifically to the chromosomal DNA across the DNA. But in these mutants, you lose the membrane binding ability. But as you lose the membrane binding ability, the ATP binding ability is not affected. As ATP is bound, this C-terminal also, these ATP bound molecules are triggered into a polymerizing mode. So the C-terminus might actually act as a switch between inhibiting this polymerization and also play a role in this conformational change, is the idea that we are tinkering with. But we need definitely more proof to say that this is an amphipathic helix that can bind to lipids. So we want to do really in vitro experiments to see what kind of lipids it binds to, take the peptide, synthetic peptide, and see if it can bind to lipid and the whole protein with just liposomes, and those are experiments that are ongoing. But currently with this, what we can at least see is that this definitely plays a role in membrane binding, and we can detect a small fraction of this membrane binding. And since these mutants do tend to polymerize, this may act as a switch between polymerization and this conversion of this molecule that is capable of binding non-specifically to the chromosomes. So they definitely affect plasmid stability. These mutations induce polymerization, and they likely prevent a switch between this para ATP bound form to a form that the para ATP that cannot bind the chromosomes. So the time delay is something like the membrane binding can account for this time delay, is something that we are tinkering with. So if a small fraction or what fraction goes to the membrane, can you get this time delay, and eventually can you get the symmetry breaking and directional movement of these plasmids? So this is the DNA segregation part I just wanted to talk about. So the modeling papers are on this Brownian Ratchet. Yes.
Very quickly, in the entire list of potential DNA segregation models, genomic DNA segregation models, I was wondering whether it was deliberate that you missed the entropic segregation model, or you don't believe it, or it's not.
I missed it, but I don't know if that should also happen, I think. It's not that I don't believe in it, but it's possible. I missed that, yes. So there's also a model that you don't need any of this active segregation thing, and DNA as a polymer, it can segregate itself into these, as the cylinders are elongating, the DNA can simply segregate into the longer cylinder. And they can just move apart themselves.
So what is the currently accepted model of DNA segregation in bacteria? Is there
It's just left at this. So at least for Caulobacter, for that particular species where the chromosome segregation has been studied, I think it is clear that DNA relay model does play a role, and it is this para gradient that sets up it. And DNA relay model is an accepted model, and Brownian Ratchet model is accepted. And even with the super resolution imaging, I think the Brownian Ratchet model is an acceptable model, but I still don't comprehend it completely where the time delay is coming. But whatever these models are, that's what it is. This time delay is absolutely essential in these models. It cannot rebind the nucleoid immediately after release from the nucleoid. How that is coming about is probably the major question there.
So there are these experiments that Anjana Badinaran has in who's at NCBS, and what she's shown is that if you induce DNA damage, then you don't get a segregation of the replicated DNA. So how would that be?
So those might be, because DNA damage might be the way, so the DNA damage might be actually forming these dimers because of homologous recombination and Recke induction and everything. So therefore you really block the DNA segregation. It's probably active signaling mechanism to stop DNA segregation. And cells also become filamentous because any DNA damage immediately induces a protein that blocks cell division. So that's why you get very long cells when you have DNA damage in E. coli. So the non-segregation of DNA might have to do more active because homologous recombination, constantly searching for the homologous, and they might be pairing, and chromosomal pairing might be happening, and then you also get entangling because they are circular. So entangling might happen and resolve base might not be functioning.
More questions? No, I'm not.
Yeah, there is some kind of numbers that were there in this paper, but I too am not very sure. But this is only, yeah, where is it? This is diffusion and simulation and, yeah, velocity of this movement of this plasmid segregation. Around 0.1 or 0.03, 0.1 micron per second. So even in this oscillating foci, they had measured speeds like 0.2, 0.3 microns per second when they are, and the oscillation are happening here. So these. Here again, but the DNA does not immediately follow the foci of the pare ATPase. So DNA is always lagging behind. That is why the filament model was proposed. It's not like the plasmid and these are not really co-localized, but super resolution microscopy really comes down to again saying that there is a gradient and plasmids are hitchhiking on the chromosome and they are bound there with the ATPase. So highest concentration where it is, that is where this.
So if there are no more questions, we'll conclude this session. And with thanks to the speaker. And maybe this is also a good time to thank the organizers, but Vijay just ran out.
It doesn't seem to be coming back.
I'm sure he will come.
And there he is.
So let's thank both the organizers for putting together this fantastic meeting, a diverse set of people. And let's thank all the speakers of this meeting for giving really nice and informative talks.